eukaryotic cell. Okay, this stuff you know. So I'm going to be pretty concise in a description of the organelles, and I'm going to just highlight what you need to know about the functions of those. Okay, so we're going to start with the first organelle. Obviously, it's going to be nucleus. So the function of nucleus, storage of genetic information. Within the nucleus, now nucleus is surrounded by the membrane, not one membrane, actually two membranes, not just a lipid bilayer, two lipid bilayers. Within the nucleus, you're going to find nucleolus, okay, which is not membrane bound, and the function of nucleolus is the synthesis of ribosomes. Okay, well, at least part of the ribosomes. Next to the nucleus, you're going to find endoplasmic reticulum. So endoplasmic reticulum, two types of it. Smooth, which does not have ribosomes on it, and it is responsible for the synthesis of carbohydrates and lipids. And rough, which does have ribosomes on it and is responsible for synthesis of proteins. Adjacent to endoplasmic reticulum is a Golgi complex or a Golgi apparatus. The function of Golgi complex is to modify, so it's modification of lipids and proteins. Also, Golgi complex packages the properly modified lipids and proteins into vesicles. And then those vesicles will be, like delivered to whatever part of the cell or out of the cell. So far, we good? Okay. A couple more vesicular organelles. Lysosomes. Very briefly, the function is digestion. Digestion of cellular fragments, digestion of food particles, digestion of whatever. We clear? Good? Okay, digestion, peroxisomes. So peroxisomes produce hydrogen peroxide. Surprisingly, eukaryotes are capable of it, and it's kind of a protective molecule there. And also, they participate in metabolism of carbohydrates. Okay, so far. Good? Moving on. Peroxisome, okay. A couple of more things in the cell mitochondria. I have a t-shirt that was given to me by students, which says, I promised to bring it to class once, which says mitochondria is the powerhouse of the cell. Everybody knows that. So ATP synthesis, right? We're going to focus on this function. It has more functions than that, but let's focus on ATP synthesis. And then we have chloroplast. Not really that super important, which contains a whole bunch of structures with the funny names like thylakoids and grana, responsible for photosynthesis. Okay. So far we good? Now let me focus your attention on these two structures here. So what's interesting about mitochondria and chloroplast is that they are products of what's called endosymbiosis. They are endosymbionts. What does that mean? These organelles used to be bacteria. Mitochondria was bacteria with aerobic metabolism. Chloroplast was photosynthetic bacteria. Eons ago, like billions of years ago, these bacteria were acquired by eukaryotic cell. Does that make sense? If eukaryotic cell acquired only mitochondria, it's a typical what's called metazoa, like protozoa or animals or fungi. If eukaryotic cell acquired both mitochondria and chloroplast, it became either algae or plants. We good so far? Now, how do we know that these are endosymbionts? Well, to be completely honest, we don't, because we formally never witnessed that moment, but sort of circumstantial evidence suggests the following. So, double membrane, both of these structures has two membranes, okay? When I say bacterial DNA, it doesn't mean that it's like bacterial DNA. It means that the DNA in these organelles looks like bacterial one. Am I clear? And they do contain bacterial ribosomes. So these organelles have their own ribosomes, which look like bacterial ribosomes. Are we clear so far? Okay? Questions here? Good? Okay, moving forward. Three more things to talk about here. First is extracellular matrix. Extremely boring thing. It's like glycocalyx. Can form capsules. Some fungi form pretty robust capsules that protect against phagocytosis. I'm not talking about membrane, because fundamentally the membrane in eukaryotes and the membrane in prokaryotes are very similar. Does that make sense? Devil is in the details. If we go into the details, there are some differences, pretty substantial differences, but for our intents and purposes, it's a membrane, okay? Now, another structure that I wanted to go over with you is cytoskeleton. So we often present cytoskeleton as the uniquely eukaryotic structure, which is not really true, because actin-like, cytoskeleton-like proteins are found in bacteria. Again, for our intents and purposes, we're going to consider it being eukaryotic structures, okay? So there are three types of filaments that form cytoskeleton in eukaryotes. Microtubules, they are, so let's go on. So what is dynamic? Dynamic means that these filaments can be destroyed and recreated. Does that make sense? Okay, so it's like your kitchen cabinets. You can remove them and put them back. It's like the walls in a house. You can move the walls and stuff like that. So, and that explains the link to the function, movement, division, associated with change, okay? Intermediate filaments are static. They maintain the cellular structure, right? If your structural elements like beams in the house are collapsing, the house is collapsing. It's not about remodeling anymore. Okay, you're pretty much boned, right? That make sense? Okay. Intermediate filaments. Hold on a second, I need to check. Okay, external appendages. Two. Flagella and cilia. Both are used for movement. And flagellum, that flagella is the only appendage that can be found in the bacteria and eukaryotes. There is a substantial difference though. You remember that bacterial flagella, they are propellers. Eukaryotic flagella are oars. Okay, they have very different structure. They're made out of microtubules and so on and so forth. Cilia can be used for movement, or they can be used to propel food towards what's considered to be kind of a mouth hole on the cell, which is really not, but you get the gist. Does that make sense? Good? Okay.